Epidemiology?
Study
Study
of
of
disease.
disease and how they spread.
What is, there are 3 basic types of symbiose. So what is a symbiose?
Living together.
Okay, living together, there are 3 types.
Mutualism, and
Okay.
reciprocity.
So, mutualism, both benefit. Commensalism, micro benefits, host doesn't really care. Parasitism, micro benefits, but the host is harmed. Basic, easy. Okay, what else are we looking at? Normally we have a lot of microbiota and flora that are in our body. They do not produce disease. These are called resident microbiota. Transient microbiota are the ones that make up part of our normal microbiota, but they're not there permanently. They're only there for, days, weeks, months. So just there's a couple examples of some of the normal microbiota on our skin: Staphylococcus epidermis or Staphylococcus aureus. Example of what the normal microbiota in the human nasal cavity looks like. Okay, so when do we acquire our natural microbiota?
At birth.
Birth. Okay, so when you're in the placenta, you're in this nice little micro-free zone, basically. And then during birth is when you start, and you keep producing for, kind of build up most of your natural flora during the first months of life. What is an opportunistic pathogen?
They don't cause disease.
Okay, so a microbe that doesn't normally cause a disease, but under certain circumstances can. So if you're immunosuppressed, so immunocompromised, if you have some kind of introduction of that microbiota to a different part of your body, so an atypical body part, or you have changes in your normal flora, so maybe they're not being kept in balance as much, so if it's allowed to overtake, then maybe it can become a pathogen. Okay? So one of the examples is tooth decay, which you can actually catch, since it is a pathogen. So, disease. Contamination, presence of microbes in or on a body, versus an infection, which is the actual invasion and growth of pathogens. So it's the difference between, is it present, or has it started actually to grow? Disease is just disturbance in our natural state of health. It's also known as morbidity. Pathogen is any kind of organism with the potential to cause disease. And virulence, it's the properties of the pathogen that promote disease. Okay, so how virulent is something? Virulence ends up being based on adhesion factors. How well does it adhere? Does it form biofilms? What kind of extracellular enzymes does it produce? What kind of toxins does it produce? Does it have any anti-phagocytotic factors? Okay. Disease conditions. Symptoms. What's a symptom?
Something we feel that you
Okay.
see.
First person, you feel it. Versus signs, which is
You see
You
it.
see it.
things that you can measure, someone else can measure. Okay? Symptoms, anything that you as the patient alone feel. Signs, whatever they can objectively be characterized. The wart was this big, the rash was this big, or whatever. Syndrome, you combine what the patient tells you with what you can actually physically see, and together it starts to characterize the disease. And you can also have some of the diseases that are asymptomatic, so right there, A, without symptoms, okay? Or subclinical. So just knowing some of that vocabulary, you can start breaking things down. How do we study or figure out what's responsible for causing diseases? Koch's postulate. Okay? So, basically you're trying to figure out, did this pathogen cause disease? If so, that pathogen has to be there in every single case. It must be isolated, grown in pure culture. When in a pure culture, it needs to be reintroduced and needs to cause the disease, and then from the diseased animal you need to be able to find that same culture, that same microbe, okay? So, find it in all of the diseased subjects, not in the healthy. Isolate single colonies, reintroduce, kills them, refind. What can't you use Koch's postulate on? What are some exceptions? What was that? I can't hear you. Ethical? True. You don't really want to do this on humans. What are some other limitations?
You can't always culture something.
You can't always culture things, okay? So, some pathogens cannot be cultured in a laboratory. Some diseases are not caused by a single pathogen. If you need multiple pathogens, then that's not something you can isolate to a pure culture because you need more than one, so it won't be pure. And then yes, we have ethical concerns because you can't really start injecting humans with things you think is going to make them sick.
Unless you're chicken.
It's not a very good idea. Developing the disease. Okay, you have an incubation period, a prodromal period, an illness, a decline, and convalescence. Okay? So, incubation. The time when you first get sick, you don't have any signs or symptoms, and this has no it varies in length. And so, you have a short period of time, you just got the disease. Prodromal period, you start feeling sick, it's usually pretty short, kind of mildly symptomatic, then you have the illness. Full bore, your body is trying to fight this off, you don't feel good, you suffer from whatever disease state it is, and you have the signs and symptoms of that disease. Then you start getting that decline phase. Your body starts figuring out how to fight that disease, starts killing off whatever is making you sick, so you start getting decline in the number of microorganisms in your system. With this decline, your body can start returning to normal function, okay, as the host destroys the pathogen. Then you have your convalescence, where, okay, your body's gotten rid of most of it, you're over the illness, and it can now start to fully repair and take care of everything that got damaged during the illness. Questions? Okay, so we're looking at systemic infections. So, entire body, systemic. So we have bacteria. So it's the presence of bacteria in the blood versus the viremia, which is the presence of the virus in the blood, versus septicemia, which is bacteria actually starting to multiply in the blood. Okay? So it's kind of like, how bad is this infection? So if we're looking at it, do we see it in the blood? Is it a bacteria? Is it a virus? Is it multiplying? Are we getting more of this? Then we have the question, how do microorganisms enter our body? How do they leave our body? The most common way of entry is usually through our face, and those openings, the most common exit is with the waist, okay? But that doesn't mean that they can't enter or exit through other means, such as insect bites or broken skin. Okay, so any kind of
waste as in
has
feces waste, right?
possible for
Waste.
microbes to go in or out. Skin. First line of defense, you have this nice epidermis with this nice thick, or depending on who you are, layer of dead skin cells that are very salty, making it a very hostile environment for microorganisms. So you have an epidermis, a dermis, and then you have more of your fat cells with the, so you have your hair follicles, nerve endings, and nerve fibers, and so you can see how, having microbes up here doesn't really give them a chance to get into this deeper part and cause infection. Or when we get cuts, why getting through down here when really you have the chances of that infection. Going through the layers, going through your defenses. Microbial infections, what do? They attach to the host cells. This is where you need the adhesion, so surface molecules on the microbe. They're very specific, They're only going to infect certain things, so this is why they need their special glycoproteins. They have their ligands. One of the methods of trying to prevent microbes is to block that ligand from being able to attach. So if you can, put something in that kind of binds to all of those ligands, all of a sudden the microbe can't attach, and you've just stopped that infection. Make sense? Okay. Anti-phagocytotic factors. Okay? So, we know that our body, part of its defense system, is trying to eat, phagocytote the microbes, and there are some microorganisms that have figured a way around that. They have caused death to our neutrophils, our leukocytes. They make it so that the phagocytosing cells are incapable. They have things that can either break down the hydrolonic acid, the connective tissue, they can break down collagen, or they can even break down blood clots. So what this looks like, you have bacteria, we have our nice epithelial cells, we have collagen layer underneath. So these bacteria can actually produce enzymes that, ACE enzymes, that can slip through the epithelial cells and start degrading the collagen. And so then all of a sudden now the bacteria can get in underneath your skin and underneath your epithelial cells, and you can start having a much deeper bacterial infection. This is much harder to treat because it's much deeper. So this is extracellular enzymes because they're making them outside themselves. And then you can have things, one of our other natural defenses is to form kind of a clot. You have these little clotting proteins. They recognize bacteria, and they, try to form a clot, kind of pack the bacteria together, make it so that they get rid of it. And ideally, this way, bacteria can't do anything, and the body has time to get some, a macrophage over there to eat it, to phagocytose it. But these bacteria, they have kinases, which could break it apart, and then they release themselves back in, so then the bacteria can dissolve the clot, releasing it back into the bloodstream. Exo and endotoxins. Okay, so toxins, anything that's poisonous. But these are produced by microorganisms. So an exotoxin is produced by the bacteria and released into the surroundings, so anything that they're secreting, versus an endotoxin, which is something that is part of them, like the, produced by gram-negative bacteria and attached to the outer portion of the cell wall. So anything that they kind of have, and so that they're toxic themselves. With the endotoxins, the classic example is the gram-negative cells. They get phagocytosed, the gram-negative cells get killed, and they release these endotoxins, which otherwise known as the lipid A, which we talked about a long time ago. So you get accumulation of that lipid A, and it can lead to the fever, the inflammation, the diarrhea, the shock, blood coagulation. Or the exotoxins, where you have bacteria, they start secreting toxins, which can actually kill some of the host cells. So they're creating a toxin to make the environment better suited for them. So if they've just, punctured that cell, then whatever was in that cell is now out and ready for them to start consuming. Okay. Endotoxin action. So this is where we have the lipopolysaccharide, lipid A, causes fever, septic shock, mobility of the capillaries, blood pressure. And it's one of those things where a lot of those endotoxins, they're heat stable. I mean, they're part of that cell wall. So you can't autoclave it. You kind of have to, makes it hard to get rid of them, but also makes it so that you have to be conscious of what kind of bacterial infection are you dealing with, because if it's gram-negative, then when you kill a bunch of gram-negative cells, you have a chance of you're going to be releasing a lot of them today. Are you ready to deal with that kind of toxic response? Epidemiology, study of transmission, spread, control, prevention of infectious diseases in a population. Big thing is population. So looking at diseases over a big population, or over any size population. You can choose your size. Communicable diseases are diseases that spread from one host to another, versus a contagious disease, which is one that is easily spread. So, you can see the difference just because is it easy to spread, or can it just be spread? I mean, there are some diseases that you have to have a needle prick for. Those aren't necessarily easy or contagious diseases. Non-communicable diseases, diseases that do not spread from hosts. Those are the ones that you can go and visit your dying relatives of because you don't have to worry about getting anything from them. Okay? Make sense? Okay, so when we're talking about the longevity and the severity of these diseases, an acute disease, acute, small, short, okay? Develops rapidly, ends rapidly. Chronic, have it for a long period of time. Subacute, it's in between acute and chronic. It's not over in a flash, but it's not there for eternity like chronic diseases. And then latent diseases, pathogens hide out for a while, and then they decide to kick on and then become active. When studying epidemiology, you gotta know about the incident rate versus the prevalence rate. So incident, how many new cases? Prevalence, how many total cases? How many are existing? How many people are infected? Versus incident, how many are new? Occurrence of the disease. Is the disease sporadic? So it occurs occasionally, it's irregular, it's, you get it in waves. Is it endemic? Stays in a population at a stable frequency. Is it epidemic? You get these sudden outbursts above typical levels, or is it pandemic? It goes everywhere. So not just this sudden outburst, but it's sudden outburst, much broader scale. Okay? So, quick example, sporadic, you got a couple. Epidemic, it's like, there's more than we're used to, but not too bad. You have your endemic, it's everywhere. Pandemic, it's really bad. Okay? So, epidemic, one burst. Pandemic, everywhere burst.
Oh, sorry.
Disease reservoirs. We talked about there's different places where the diseases can hide out, where these pathogens can hide out. Where is that source of infection? Humans can be nice reservoirs. We can be symptomatic or asymptomatic, so we may or may not show that we have a disease, but it still means we could pass it on. Animal reservoirs, zoonosis, so a disease that exists primarily in animals, but can be transmitted to humans. Usually in those cases, we are the final carrier. And then we can also have environmental reservoirs, like soil and water. So this was an example of cholera in 1854, so you can see, being able to actually look, okay, how many people are dying? Where are they dying? And then you start laying in what's causing them. In this case, you had contaminated water. So you're being able to see that, oh, okay, right here's where it's coming from. It's from these pumps. So, ways of this is that you can get spread of diseases. You can get it through some kind of medium. Do you want water, food, or air. Waterborne transmission, you could, a lot of times it's the indirect, so you get that oral fecal transmission via contaminated water, cholera. You can have airborne transmission, which is the most common route, so the common cold, just, it's in the air. And you can also get the foodborne transmission, so improper handling of foods, when you get food poisoning, tapeworm. You can have direct contact transmission. So you directly contact someone, the touching, the kissing, the sexual intercourse, direct contact to get the disease. There can be indirect contact. So this is the foam I teach. You guys are going to be gram staining today, seeing what things you found on non-living objects that can harbor and transmit the pathogens. So you'll have plenty of ideas of what those are. And then droplet transmission. The disease agents are spread by droplet nuclei. So droplet transmission.
Wait, can you go over, can you go back one more? What's the difference between vehicle water transmission and waterborne transmission?
Vehicle transmission, just saying that, you're saying that these are being carried by something, which is the same, I mean, because vehicle just means it's being carried by something. It's either being carried by water, it's being carried by food, it's being carried by air. I mean,
So
this is kind of like the big category that all of these are in, because, I mean, waterborne, airborne, and food.
Oh, so waterborne is a part of vehicle transmission?
So is air and food. They're
Oh,
all
okay.
vehicle, because they're all being, you know, there's some kind of vehicle that's transporting this to you.
Oh, okay.
Make more sense?
Yeah.
Okay. Okay. Living organisms can transmit diseases to humans, so we can have mechanical transmission, so passive, so transmission of pathogens or body parts, or you can have biological transmission, so the active transmission, transmissive pathogen. And it must complete part of its life cycle in a vector before infecting a human host. Okay, so with that biological, that's where we get some of those mosquitoes. They have to, some things have to harbor inside mosquitoes before they can infect us. Other times, passive pathogen just kind of, you know, moves from person to person. It's like, oh, I'm good, new host. Okay. So, flies, very bunch of diseases. EID, emerging infectious diseases. So, the EID, they're looking for changes in disease. What is the increase rate? Do they have, are we having increases in, you know, in incident? Is something becoming more virulent? Is there some new horrible thing coming to get us? So the CDC, the WHO, and the NIH are all monitoring and controlling, looking for emerging infectious diseases. They're all trying to figure out what is going to be the next emerging infectious disease and how can we stop it or prevent it or slow it down when it does come. Okay. There are a bunch of notifiable diseases. These are diseases that physicians are required to report to the U.S. Public Health Services. And then there's the MMWR, so morbidity and mortality weekly report. So we're looking at what is the incident of a specific non-notifiable disease and how many deaths are we getting from this notifiable disease. And so they keep constant records of, okay, anything that the doctors have to report, you know, we don't just want them to report it and nothing to happen. We have these weekly reports that are made up so we can keep track of all of these, you know, notifiable diseases. So we know how many instances are we dealing with, how often, how many people are dying, what's going on with this. Okay. So, hospital-acquired infections. These are usually opportunistic pathogens. Exogenous infections, they're acquired In the healthcare environment, usually that's where you're going to get the ones that have resistance to a lot of different antibiotics because they've been circulating around the hospitals for too long. Endogenous infections, these are acquired from normal microbiota within the patient. So if your normal microbiota now, because you have some kind of fluctuation or something has changed, so it can now become an opportunistic pathogen. What were the things that can make it opportunistic? What are the circumstances? Compromised immunity. What else?
Changing the patient's microbiota.
Change your microbiota. Or, so if your microbiota changes, then that means that it can maybe suddenly grow out of control. It's not being kept in check. What would be the other thing?
Introducing a new microbiota.
Well, yes, but that would be exo. Talk about endo. They're becoming opportunistic pathogens. You take something that is in you and move it to a different part of your body. So if it gets to transfer body parts or body, cell types, so then it could become an opportunistic pathogen. Some of the microbes in our intestines would be kind of like they get kept in check because of our stomach acid. If they get out of our stomach, then we start having issues. Okay. And the iatrogenic infections, they're acquired from medical procedures. So the thing is, you are physically taking some kind of object and putting it into a patient. And with that, you could be carrying, catheters, you could be carrying microbes into a person. Or if you cut them open, if you're actually cutting them open, you're putting a scalpel and you're putting, we do a lot of things to try and make sure all that's sterile, but you're putting things physically into the patient. There is a chance that they could get an infection. Or you're just opening them up so that you get an infection. Yes.
What does the endo kind have to do with being in a hospital? It's already in your body.
Yeah, but usually you're in the hospital for a reason. So if you're in the hospital, you're getting treatments. If you get, that could be why you're compromised, or that means you could be taking a drug that messes with your normal flora, which would then allow it to be opportunistic. So if you're in a hospital, you're probably taking heavy duty drugs, which are going to mess with your normal biology.
Thank you.
That make more sense?
Yes.
So the factors: presence of microorganisms in the hospital, immunocompromised patients. Also, you have to worry about the transmission of pathogens between staff, patients, and among patient to patient. So those are all things you have to worry about that make these infections so that you end up getting more and more sick in the hospital.